is a very important concept. Most important I want you to get from this. Yes, genetic transfers, conjugation tubes, or sex pillars, or conjugation pillars, it's all the same. It allows genetic transfer among species, or even in species. Most important thing, though, when it relates to the clinical environment, this is one of the major causes for antibiotic resistance. Why? Because genes can be passed. These are genes that code for resistance factors that can be passed from one cell to the other. All right? Okay, the next structure is a flagella. And again, all I want you to remember is just the basic function. Don't memorize all of these different structures here. A flagella, why is that so significant? Well, a flagellum allows an organism to move from one place to the next. So motility is big. Think about a sperm cell. A sperm cell has a flagellum. Bacterial cell also has a flagella. It allows an organism to move. It allows chemotaxis. So chemotaxis, think about the term chemochemicals, taxi to move. It allows a bacterial cell to swim from one area to the next. And it's also involved with attachment. So flagella can attach to tissues. There are many different types of flagella bacteria. How we classify them very differently. Let's look at the terms. And by the way, this is just an illustration, right? Monotrichus, one, has one flagellum. Amphi on both sides, half and half. Right here, these are the flagella. Lopho on one side, that's Lophotrichus. And then peri means around. This is a peritrichous organism. Here's some real slides. These are real organisms. This one here looks like that one, right? So this bacterium has a Lophotrichus flagella. Whereas this one, take a look at this one. The dark stuff is the bacterial cell, all these little strings of the flagella around it. That looks like this one here. So this is peritrichous flagella, right? But nevertheless, here's what I want you to know. What's the function of flagella? Motility, all right, chemotaxis, and then attachment. That's all I want you to get from this slide. All right. I just showed you bacteria that have flagella. Remember I mentioned to you spirochetes. These are spiral organisms, corkscrew-like. They also have flagella. But instead of what we saw here, let me go back to that slide. These flagella are on the exterior part of the organism. Spirochetes have a flagella inside the cell. So this is called an endo. Endo means inside, an endoflagella. So think about this flagella moving. It's going to cause this organism, this bacteria, to twist like a corkscrew, actually. So that's what it looks like here, all right? Spirochetes have an endoflagella. Now, the other structure I'd like you to know is the glycocalyx. All right? Let's kind of decipher the term. Glyco means sugar. Calyx means a coat. So it's like a sugar coat. It's not just carbohydrates, by the way. We can have lipids in there. We can have small proteins in there also. Now, there are two basic types of glycocalyx. And they're illustrated here. At least one is. The first one is a capsule. A capsule is a pretty thick glycocalyx. A slime layer is pretty thin and diffuse. So let's go to the slide here. You notice this is the organism that causes anthrax. It's called Bacillus anthracis. Notice how snotty that is? It looks like a little snot on the plate. If you were to stain this, that snotty stuff is what you see here in green. That's the glycocalyx. The black area back here, that's a bacterial cell. It's a rod, of course. So the rod is here is producing this very thick glycocalyx. That's called a capsule, all right? So a thick glycocalyx is called a capsule. A thin or diffuse one is called a slime layer. I don't have an example of a slime layer here, but nonetheless, I do have an example of a capsule. So again, glycocalyx is supported around the bacterial cell. It can be pretty thick. That would make it a capsule. If it's pretty thin and diffuse, this would make it a slime layer. So the only two I want you to know here is this one. Bacillus anthracis, this green stuff that you see is poly-D-glutamic acid. This is what it's composed of. The other one is this one here, Streptococcus pyogenes. This is the organism that causes strep throat. It causes scarlet fever. That glycocalyx, right, you can see it's not as thick as this one, but maybe you can see the bacterial cell outline and then the calyx around it. That calyx is made up of hyaluronic acid. All right? So hyaluronic acid for Streptococcus pyogenes, poly-D-glutamic acid for Bacillus anthracis, the organism that causes anthrax. All right. So moving on. So here's what we talked about so far. Pili, just to let's sit back for a while. Make sure that we're comprehending what I'm trying to get through here. Basic focus of this chapter is on some bacterial structures. So we talked about pili. There were two types, primary, and then we had the sex pillars or the conjugation pillars. Then we talked about flagella. All right. We just talked about glycocalyces. And now I want to talk about the bacterial cell walls. And again, all of this stuff, well, the cell wall stuff was talked about in the laboratory last week. So if you recall, most bacteria have cell walls. There are a few that don't have cell walls. But if a bacterium has a cell wall, the basic ingredient, this is called peptidoglycan. Your cells don't make this. Fungal cells don't make this. Parasitic cells don't make this, but bacteria do. So this is unique to bacteria, right? So when bacteria have cell walls, like you see here in blue, the basic component is called peptidoglycan. Now, of course, I just showed you a gram stain. We had gram positives that were blue and gram negatives that were red. The reason why they stain differently is because they have different things in their cell walls, in addition to peptidoglycan. All right? So gram-positive bacteria, they have peptidoglycan and they have teichoic acid. Remember that. Gram-negatives have peptidoglycan, but they don't have teichoic acids. Let me just show you some slides back here. As an example of a gram-positive organism. This brown little thread, like you see here, that's peptidoglycan. If you know that, that's pretty thick, right? So gram-positive organisms tend to have a very thick peptidoglycan layer. The other structure I want to point out, these little purple ones, that's teichoic acid. You don't find teichoic acid in gram-negative organisms. So just to summarize this slide, here's what I want you to know. Gram-positive organisms have peptidoglycan right over here. All right, that's that structure. And they have teichoic acid. What about gram-negatives? In fact, let's just go back to this slide. You see how thick this layer is? Those brown strings, that's peptidoglycan. Take a look at gram-negatives. Not as thick, right? So gram-negative organisms, here's what I want you to know. Yeah, they have peptidoglycan, but it tends to be thinner when compared to gram-positives. So this is peptidoglycan. They don't have teichoic acid, but they have this lipid type of molecule above the peptidoglycan. That's called lipopolysaccharide, LPS. So polysaccharide, of course, is carbohydrates, but it has also lipids. It has lipids in there also. So we have lipopolysaccharides made up of polysaccharides and then that lipid. That lipid A, don't forget, this lipid A, right over here, is part of lipopolysaccharide. I do want you to know this too. Another name for lipid A, don't forget it, is called endotoxin. All right, so lipid A and endotoxin is the same thing. This is one of the components that's responsible for something called septic shock. All right, but right now, here's what I want you to know. Gram-negatives have a very thick peptidoglycan layer. We have LPS, lipopolysaccharide. Part of lipopolysaccharide is lipid A. Lipid A is also called an endotoxin, not an exo, but an endotoxin. All right. Now this slide, you're gonna like this. You don't have to know anything from this slide. What did I say about archaea? Archaea don't cause disease in humans. So we're not gonna study this slide here. All right, some other things just to tie this up. Other structures that we see, membranes, cell membranes. Many different things are on the plasma membrane of a cell. First of all, it separates the cytoplasm from everything away from the cytoplasm. So it's a nice little barrier. It's a boundary. Transports many chemicals, including amino acids, sugars, to that membrane. There are enzymes on the membrane, on this plasma membrane. So again, many different things are associated with the plasma membrane, not just one thing. There's water in there. There's nutrients in there. Amino acids, I just mentioned them to you. Proteins, sugars, salt, vitamins, ions, nucleotide bases. Sometimes what you would find in the cytoplasm are these little structures here. They're called inclusion bodies. All right over here, inclusion bodies. And again, a way to illustrate what these are, I'll give you an example for your system. When you eat too much glucose, what happens? Your liver cell can take the excess and form something called glycogen. So glycogen is a polymer of glucose. That's a nice energy reserve for you, right? So you imagine you're in the desert for a few days, you run out of energy. What happens? Your liver starts cutting up glycogen into glucose molecules. And now you can send glucose to the bloodstream. Bacteria are not that stupid too. If they have too much of one thing, they take those things and kind of pile them up for later use. So again, inclusion bodies are simply this. It's basically a nutrient reserve, and there are many different types of inclusion bodies. For example, glycogen, there's sulfur, lipids, polyphosphate, even magnetite. So magnetite is iron sulfate. We don't understand why bacteria have them, but again, it seems to orient the bacteria to the poles. We don't understand why. All right? So again, here's the cytoplasm, many different things in there. You have a DNA that's called a nucleoid. You have these plasmids, and then you can have all of these other things here: sulfur, glycogen, lipids. Not all bacteria have all of them, by the way, these inclusion bodies, but many do. All right. Let me give you an example. This is really taken from a true bacteria. This is the bacteria that causes diphtheria. It's called Corynebacterium diphtheria. So these, of course, are false colored. So the green is the rod. You see this little purple dot back here? That's called a metachromatic granule. What it is polyphosphates right over here. So that's polyphosphates. The point is this. Bacteria can take things that they have in excess and accumulate them in their cytoplasm into what we call inclusion bodies. They're not just junk. The bacteria is going to use that as a resource later on and starts replicating itself. All right? Let me just wind up by talking about plasmids again. Plasmids are very small pieces of DNA. They tend to be circular molecules. They're extrachromosomal, meaning the bacterial cell doesn't need this. The chromosome or the nucleoid, it is the genetic blueprint. Plasmids, just by chance, bacteria can have them. It's not required for the survival of that organism, given a natural environment. But let me just say this, the most important thing is this right here. Plasmids can contain genes, so this is DNA, contain genes that code for resistance to antibiotics. And that's the big problem that we see, all right? Okay, other things you find in the cytoplasm, ribosomes. You guys should all know this by now, ribosomes. These are the structures on which proteins are synthesized, all right? Other types of subcompartments we see in the cytoplasm, gas vesicles, kind of unique. So this is a bacterial cell. You see all these little stacks back here? They're like little bubbles, but it's organized very specifically. And again, this is a tremendous feat for bacteria. Think about this. Remember we mentioned cyanobacterium, they were photosynthetic bacterium. Gas vesicles are typically found on bacteria that photosynthesize. Why? Well, imagine there's a bacteria living in some pond. If it sinks to the ground, right, it can't see light. So basically it can't photosynthesize. But you put gas vesicles in them, like what you see here, they float to the top. They're pretty tricky, actually, for these simple organisms, right? But not so simple after all. And now they have direct access to sunlight. So E. coli wouldn't have this. Staph aureus wouldn't have this. Other bacteria that cause disease in humans don't have this. Why? Because that's not the environment these organisms live in. So again, some organisms have gas vesicles and allow them to be buoyant so they can be on the surface. So these are primarily for the photosynthetic ones. Other ones you can see, other bacteria have magnetosomes. I just mentioned iron sulfate. It allows the organisms to orient themselves to the poles of the planet. We don't understand why. All right? Guys, that's a very basic review of some really important structures. Again, glycocalyx really important. We talked about the pili, very important. Structures like those. All right? Do you guys have any questions for me?
Excuse me?
Yeah?
So for the quiz on Wednesday, it's over chapter 1 to the one that we have today.
Exactly. And again, I want you to, I don't want you to be nervous. This is a really simple quiz. And it covers some very basic stuff. I think I mentioned to you, I don't think I have any of those questions, really not questions. They're just little statements or little phrases. That's more than 6 words, right? And just to give you a hint, we talked about definitions: anaerobe, aerobe, cyclophile, facultative, right? First chapter, we talked about some individuals. I kind of labeled them for you, like Robert Hooke, Pasteur, Fleming, just a little blurb about them. Leeuwenhoek. And again, it's just true, false.
For those, for the scientists, we don't need to know like the dates. Right?
No. So that's a good question. Don't waste your time memorizing dates. It's not a class in history, but I do want you to know the significant things they've done.
Okay.
All right? Good.
For the quiz, also, will we need to download Respondus?
No. So